In this screencast, we are going to start by talking about what happens when there are multiple alleles involved in genetics. So many genes are going to have two or more alleles that are said to have multiple alleles. And the best example for a problem involving multiple alleles is going to involve the coat color in rabbits. And coat color in rabbits is actually determined by a single gene that has at least 4 different alleles. And these 4 alleles demonstrate something called dominance hierarchy. Miko, stop barking. Which means that there are some alleles that are dominant over others. So the 4 alleles for coat color in rabbits are as follows. They could have a capital C, which is representing the full color, sometimes also called wild type. We can have a allele that is represented by a little c with a CH up above, which is representing a light gray or chinchilla. We can have a little c with a little h, which is going to give us an albino rabbit, but it's going to have black extremities. So its ears, its nose, its paws will sort of be black. And then last but not least, we can have an individual that is just albino, represented by a little c. Okay, so these alleles here are listed in the order of dominance, and so we can show the different possible genotypes of each of these rabbits. So full colored rabbit, it could have a big C, it could be big C, little c, CH with a chinchilla, it could be big C, CH, or it could be big C, little c. All 4 of those would give us the full colored rabbit. Then for the chinchilla colored rabbit, it could have the allele, 2 alleles for the chinchilla color, making it the chinchilla color. It could have 1 for the chinchilla color and 1 for the albino with black extremities, or it could have 1 allele for the chinchilla color and 1 for just plain albino. Then the rabbit that is albino with the black extremities, also known as a Himalayan rabbit, it could be CH, or it could be CHC, and just have 1 allele for the albino with black and the other as just plain albino. And then last but not least, the albino, its only possibility is going to be little c, little c. So let's talk about an example here. If we were trying to find out what types of offspring could be produced by a full color rabbit that had a genotype of big C, let me rewrite that, with little c, CH, and it was bred with a Himalayan rabbit, right? That was the one that was the albino with the black extremities. That was little CH, little c. So there's our Punnett square. We're going to put these alleles up here and these here. Again, I have capital C there showing the difference between uppercase and lowercase, and I have that in cursive. So take a second here to pause and write in what you think will happen here in our Punnett square. And this is what you should have. Then go through and find our genotypes and phenotypes. Possibilities there are going to be capital C, little c, H, little c, CH, CH, and big C, little c, and C, CH, little c. Count out the numbers there. We have 1 out of 4 of each. And if we're looking at different phenotypes, 2 of those 4 are actually going to have the full color. Those are going to be the ones with a big C, right? So here and here will represent the full color. And then the remaining 2 are out of the 4 are going to be chinchilla, or the light gray. And we know that chinchilla will be represented with any of those little C's. So here, this one here and this one here will be our 2 out of 4, giving us the chinchilla or light gray color. Okay, so let's move on now to talk about something called polygenic inheritance. And this is when there's going to be one trait that is affected by the expression of many genes. For example, people, they can vary a lot in traits like height and nose length because there are lots of genes that can affect the growth of a person. So the determination of a given characteristic is going to be the result of the interactions of many genes. So this image here is of a bunch of different contact colors, but eye color is one of those things, along with size and height, shape, weight, those sorts of things, where you have many genes accumulating and creating a combined effect. So we call that polygenic inheritance. So a trait that's going to be affected by a number of genes, or polygenes, it's not going to show a clear difference between groups of individuals. And so instead it shows a graduation of these small differences. Many of the normal human traits we think are polygenic, right? So like hair color, eye color, skin color, that sort of stuff. Another important thing that can happen with genes is something called epistasis. And this is when the expression of a gene will be controlled by the expression of another gene. So for example, in mice, there are going to be different genes for fur color, but there also is going to be a gene that controls whether or not any pigment is produced at all. So you can have mice that have genes for black and something called agouti colored, meaning that the coloring will be banded in their fur. But if you have the pigment gene missing, then they all are just going to appear white, and we call those the albino mice. Oftentimes the genes are going to be shown in a dihybrid cross because they're really closely related and they'll affect each other. They'll be affected by the location on the chromosome. We will see these in examples that the class goes over when we talk about the infographics a bit more. And also in the infographics that you created, we will talk about pleiotropy, and this is occurring when one gene will control many traits in an animal. So, for example, the gene in pea plants that will express either a round or wrinkled texture in seeds will also influence the phenotypic expression of things like how it's able to absorb water and its metabolism of starch. So again, more on those when we go over the infographics, and we'll talk about specific Punnett squares, but I just wanted to introduce them to you here. And finally, I want to end by talking about sex-linked genes. There are many genes that we find on the X chromosome, but we know that the Y chromosome actually doesn't contain a whole lot of genes. We think it only contains a few, and most of the time it's related to the production of sperm. So since the X and Y chromosomes are determining the sex of an individual, all of the genes that we find on those chromosomes are said to be sex-linked. Scientists have found more than 100 sex-linked genetic disorders that are now associated with that X chromosome. A couple of examples here for you are color blindness, hemophilia, and muscular dystrophy. Hemophilia is going to be a disorder where the blood isn't clotting normally. And each of these examples are caused by recessive alleles. Since males only have one copy of the X chromosome, they are only going to have the disorder if they inherit just one copy of the allele. But for females, they have to inherit 2 copies of the allele, one on each of their X chromosomes, for the trait to show up. So sex-linked genetic disorders are going to be much more common in males than females. Let's go through just an example here for you. We'll do it for hemophilia. So let's look at the different genotypes, and we'll represent it with H's. But remember, these are the sex chromosomes, so we're going to use X's and Y's. So X, and then it can have an H associated with it, 2 capital H's. That would give us the normal blood clotting female. Then females could also have a big H and a little h. So that's going to again be a normal clotting female. But there'll be a carrier of this hemophilia, as we can see with this allele. Then we could have a female that has the 2 little h's, and that female will be a hemophiliac. And then getting to the males, we know that they have both X and Y chromosomes. The X could have a capital H associated with it. We know the Y isn't going to carry anything. So the X, this is going to be a normal blood clotting male. And then its X chromosome could have a little h associated with it, meaning that it will be a hemophiliac. So let's do a practice problem here and say that we have I'll go to the next slide. Let's say we have a normal woman. Her father, he had hemophilia, but she married a male that was also normal. And we want to know what is the chance of hemophilia in their children. So let's first find the genotype of the woman's father. If we go back and look, we know that for males, if they are hemophiliacs, the only possible genotype they can have is right here. Then let's find the genotype of the woman. We know that she inherited an allele from him that had the hemophilia on it, so we'll know that she'll have at least 1 little h, but she won't have 2 because she herself doesn't have hemophilia, so she'll have a big H allele. And then let's look at the genotype of the man. He, we know, is not hemophiliac, so he's going to have the normal capital H allele. So there we go. And now we're going to do a cross of these two. We'll put the female's alleles up here, and then the male's X, H, Y there. Go ahead and pause and fill in this chart on your own first, and then see if you get what I get. And so again, you're just treating each of these as separate alleles and just combining them in the boxes the way we normally have. Let's go through our possible genotypes and phenotypes. We can have a X capital H, X capital H, like we have there. We can have an X capital H, X little h, like we have there. X capital H, X little h. We can have a X capital H, Y, which we see here. And last but not least, we can have an X lowercase h, Y. And we have a quarter for each of those. But when we get to our specific phenotypes, we know that this one up here, this is coding for a normal female. We know that this is coding for a normal female. This here will give us a normal male, and this will give us a hemophiliac male. And if we count it out, we'll have half of them be normal females, a quarter of them will be normal males, and 1/4, as we can see here, has that little h allele, meaning that this male will be a hemophiliac. And let's do one more example here. The gene for color blindness is carried on the X chromosome, and again we know that it's recessive. So if a man whose father was colorblind has a colorblind daughter, we can figure out if the man himself is colorblind. We know his father will have to carry the colorblind gene. This will be his genotype. And because we know it's recessive, that means the daughter has to carry both of the colorblind genes. I'm designating this with little c's here in those 3 instances. If it had a capital C, I would make it cursive. But if the daughter had X big C, X little c, she would still have normal vision. She would just be a carrier of the colorblind allele. So we know that she has to have both of the colorblind alleles. And we know that the man is colorblind because the colorblind daughter had to get one of her genes for colorblindness from her father. So he will have the same genotype as his father. So where did the man that we're talking about here, where did he get his gene for colorblindness? A man is going to get his gene for colorblindness from his mother, right? And then he gets his Y chromosome from his father. And another question for you, must the fathers of all colorblind girls be colorblind? And try to explain that for me. So actually, yes, all fathers of all colorblind girls are going to be colorblind themselves. For a girl to be colorblind, she has to inherit the colorblind gene from both of her parents. So that ends the screencast here, but here are the 5 main things that we mentioned in this screencast, so make sure that you're comfortable with what each of those situations looks like.